And welcome everybody to the Pathology Grand Rounds for the November lecture. Can I remind you that the Pathology Grand Rounds are co-sponsored by both the Jean Shanks Foundation and the Pathological Society. For members of the audience, if you have questions that you wish to ask, please click on the Q&A box at the bottom of your screen and enter your question there, and we will address the questions at the end of the lecture. Now, it's my great pleasure to introduce today's speaker, who is Raghav Sundar. Raghav is a clinician-scientist specialising in gastrointestinal oncology. He's a consultant medical oncologist at the National University Cancer Institute, Singapore, and the research director for the Department of Haematology-Oncology at the National University Hospital there. He treats patients with gastrointestinal malignancies, including colorectal, gastric and hepatobiliary malignancies. His research interest is in genomics and epigenetics of gastric cancer and tumours that metastasize to the peritoneum. He studies the interaction between the tumour epigenome and the immune microenvironment, and that is the theme of his lecture to us this morning, which is entitled Epigenetic Interactions Between the Tumour and Microenvironment. Thank you, and over to you, Raghav.
Thanks, Mark, for the very kind introduction. And I'd like to thank the Society for inviting me to give this talk. I'm going to share screen now. Is this visible to everyone?
Yeah, we can see that.
Yep, that's fine. Thank you.
Okay, very good. I'll get started then. These are my disclosures. So today during my talk, I'm going to talk a little bit about some paradigms that we are hoping to shift as medical oncologists. And I'm going to talk about how we are shifting these paradigms. And one of the paradigms that has been entrenched into me as a medical oncologist and even as a medical student or as a trainee is this idea that to treat cancer, one needs to kill cancer cells. And when I was in medical school, when I started my medical oncology training, we were taught these very complex metrics on how the tumor cells will grow over time in an exponential fashion, and how the different interventions that we give to the cancer will actually kill the cancer cells, including things like surgery and chemotherapy, and why we needed to give chemotherapy in cyclical fashion so that you can hit the cancer cells at various stages of its mitotic cycle, depending on the mechanism of action of the chemotherapy in order to kill the cancer cells. And we were then taught all these different models on the way the cancer cells grow, and why these different models then explain the way different chemotherapy doses, higher doses, combinations of chemotherapy would work. But at the end of the day, we all know that chemotherapy only has a very limited effect on the cancer cell. And today I'll share a little bit with you about why this is so. And so at the end of the day, the truth is that medical oncology treatment in the 20th century was pretty much just Hulk smashing the cancer cells with chemotherapy. And this was a little bit also because I need to go through a little bit of the history about how we understand cancer. And by understanding how we understand cancer, we get a better idea of some of the new and emerging concepts that are coming out, and how we are using these concepts to identify potential biomarkers and therapeutic targets. And so to give you an explanation of this, I'm going to use the Singapore's mass rapid transport system map. It wasn't as complex. It isn't as complex as the London underground rail system, but over the past 5 or 10 years, it has become pretty complex. When I first moved to Singapore 20 years ago, we only had the green line and the red line. But this is essentially what the rail transport system looks like today. And if you think about it, in order for a person who starts in this railway station over here, if he needs to get to this railway station over here, or this train station over here, they need to actually navigate through many different intersections and train lines and understand how these happen in order to be able to navigate through this railway system to get to their destination. And if you put this in the context of now cancer and how we understand the map that has been laid out, and if this is indeed cancer, then this will start to give us a little bit of a better idea of how we treat these cancers. And this is essentially chemotherapy. So if you nuke the entire railway system, then there's no way that the cancer is going to be able to get from here to here. And that's how we kill cancer cells. But we have started to understand a little bit more about how cancers, we have started to understand a little bit more about oncogenic pathways, and by blocking these pathways that we can actually induce cancer cell kill. And this essentially is, for example, us understanding the fact that this pathway, for example, is more utilized by a certain tumor to get from here to here. You could imagine that the same person would choose to go through another pathway to get to here, but he is indeed choosing this pathway. Then by blocking this pathway, you would induce cancer kill. But as you can see in this figure now, you realize that most of the time for us to understand this, to identify these pathways that are being upregulated by the cancer, most of these are identified using DNA sequencing. And in DNA sequencing, one of the problems that you have is that you only understand the pathway that the cancer is utilizing or upregulating. You don't actually understand what's happening around this. And one of the problems, and I mean, this essentially brings us to the era of targeted therapies now, where by identifying pathways that cancer cells are using to grow, and by blocking them, you would achieve cancer kill. But one of the problems with a lot of the targeted therapies that have been approved for use in medical oncology treatment is the fact that most of these targeted therapies eventually will acquire mechanisms of resistance to the treatment. And this essentially, if you can see, this is a study that looks at alectinib after progression on first-line crizotinib for ALK-positive lung cancer. And you can see that although there is a survival benefit for alectinib over chemotherapy, that eventually there is all patients will still die with targeted therapies, especially in a metastatic setting, which means that eventually the cancer does still figure out a way in which it can grow, even with targeted therapies. And that brings us finally to the tumor microenvironment, which is going to be the focus of our talk today. And with a greater understanding of the tumor microenvironment, then we start to realize that there is a lot more to cancer than just the cancer cell itself. And by just using drugs or understanding how the cancer works as an entity without understanding the way the cancer interacts with the tumor microenvironment, we aren't going to be able to break the next frontiers and barriers of cancer therapies. And therefore, starting to focus on the tumor microenvironment with things like such as the tumor vasculature, as well as the immune cells that surround the cancer cells, such as the dendritic cells, T cells, tumor-associated macrophages, as well as the surrounding stroma, which includes the extracellular matrix, as well as the cancer-associated fibroblasts. We do know that now that the emerging therapeutic targets that we are identifying, the most famous that has been brought into date would be immune checkpoint inhibition, is only the tip of the iceberg of what is likely going to be a barrage of new therapeutic targets that are going to appear in the next few years because of our understanding of the tumor microenvironment. And so that's kind of why the platforms and the assays that we use to study the tumor has to go beyond DNA sequencing into understand, because with DNA sequencing, you're only going to be able to study the tumor and not the microenvironment. And therefore, we need to now develop platforms to study the tumor microenvironment. And therefore, the questions will start to arise on things like how much of information about the tumor microenvironment do we need? Do we need to just know the location of the microenvironment with respect to the tumor? Like, for example, looking at the immune cells in the invasive margin versus immune cells in the tumor stroma? Does the distance between the cells matter? Does the distance between the tumor cells and the immune cells, does that matter? Does the distribution of the immune cells matter? Whether they are in a compartment or whether the spatial proximity makes a difference, does that matter? Or are there special patterns that, special spatial patterns that matter in this? For example, your tertiary lymphoid structures or pre-metastatic niches such as the brain, the bone, the peritoneum. So the question is, how much of information of the tumor microenvironment do we need, and how do we obtain this? And so I'm going to talk to you a little bit about how we are starting to obtain this information. And traditionally, the most the simplest way in which we started to understand the tumor microenvironment was using bulk RNA sequencing. And so in bulk RNA sequencing, again, going back to the Singapore train map, the way we do bulk sequencing is essentially we take a bit of the tumor, we grind it up, we extract the RNA, and then we perform sequencing on this. And one of the issues with performing bulk RNA sequencing is, yes, you will understand a little bit about the tumor microenvironment, because at least you will start to understand that, okay, there are certain proportions of cells beyond the tumor cells that exist within the bit of tissue that we've sequenced. And that would be, you will say, okay, there is a proportion of T cells and B cells and maybe some stromal cells that are present. But beyond just the proportions of cells, you don't actually understand more about the tumor microenvironment without, from just bulk RNA sequencing alone. And of course, to be fair, now that I'm talking to a pathology audience, the pathologists actually have a much better understanding of the microenvironment just by looking at it under H&E or under specific situations. But from a sequencing point of view, bulk RNA sequencing actually only gives you a very little insight into the microenvironment. And that's why we have moved on to single cell RNA sequencing now, where beyond just grinding the entire tumor up, usually for single cell RNA sequencing, what we do is the tumor is first dissociated into single cells, and these are bar-coded, and then the single cells are then sequenced individually. And this now starts to give you a little bit more granularity and understanding of the microenvironment, because apart from cell proportions, you also now start to understand cell states. And I will talk a little bit about cell states and the importance of understanding cell states from the single cell sequencing data. But this cell states, if you broadly put it back into the context of the train system, is apart from knowing that there are just a few different colored train lines that exist, you start to understand perhaps a little bit about the different train stations that exist within the train lines that you see through single cell sequencing. But of course, the latest in this entire range of platforms is spatial profiling, where beyond just individually dissociating the single cells and then sequencing them, if you retain these cells on in their geographical platform, in their geographical state, and then profile them while they're retained in this state, then you actually start to understand in a much more deep manner the spatial, the association of the spatial, or rather the importance of the spatial associations with the genomic patterns that are coming out from the sequencing. And this is essentially the latest technology that are emerging. There are many different ways of spatial profiling. And I use the word spatial profiling here because while you can retain the spatial orientation of the tumors on the slide before you either perform transcriptomic sequencing, but you also can perform multiplex immunohistochemistry using multiple proteins or other sorts of spatial profiling to understand different aspects of the tumor while maintaining their geographical orientation. And this is essentially kind of a lot of what we are currently doing in our lab, and spatial transcriptomics is truly where the genomic biologists such as myself would work very closely with the pathologists to identify specific orientations or areas of the tumor that are worth studying using spatial transcriptomics. And I'll share with you a little bit about the data that we have generated over the past couple of years. And for the rest of my talk, because I'm a gastric cancer oncologist and my research is in gastric cancer, I'm going to focus, share with you a little bit about the work that we have done in this space in gastric cancer. And we know gastric cancer is actually a very heterogeneous tumor. There is a lot of inter as well as intra-tumoral heterogeneity, which means that there is heterogeneity between patients in the gastric cancer samples, but also within the tumor itself. So there is heterogeneity between the primary tumor as well as the metastatic lymph nodes, which may be different from the distant metastases. And this heterogeneity actually changes with therapeutic pressures. And a lot of the precision oncology ventures that we have been pushing in this space actually has to address this issue of this heterogeneity that exists in gastric cancer. And one of the issues with this heterogeneity that we see from a clinical relevance point of view is the fact that most patients are diagnosed with gastric cancer when they either present with abdominal pain or dyspepsia or with a bleeding GIT, and then they get an endoscope done where a stomach tumor is identified, and the gastroenterologist or the surgeon will take a biopsy of the mucosal aspect of the tumor and send this off to the pathology team to read it. And then usually once you suspect cancer, you will send the patient for a CT scan. And very often when a CT scan is done and the CT scan shows that the patient has locally advanced or metastatic disease, then during the multidisciplinary tumor board meetings, the patient will be discussed and said, okay, this patient will benefit from systemic therapy and is referred to the medical oncologist. And then the medical oncologist says, okay, let's perform biomarker testing on the gastric cancer sample. And very often the biomarker testing is then performed on that mucosal biopsy sample. And so one of the questions we asked is, does the superficial layer of the aspect of the tumor or the mucosal aspect of the tumor that is biopsied by the endoscope, is this different from the deeper aspects of the tumor, the serosal aspects of the tumor? And are those different from the metastatic lymph node? And so in one of the first few studies that we did addressing spatial heterogeneity in intratumoral spatial heterogeneity, we collaborated with Professor Heike Grabsch, who's in the audience and invited me to give this talk. In this study, what we did is from 60-odd gastric cancer patients who had undergone surgical resection of the gastric cancer samples, Heike actually manually annotated all these samples into 3 different regions. So there was the superficial region, which was actually divided into 2 parts. We had 2 superficial regions as well as 1 deep region, and then the metastatic lymph node. And based on this, there were at least 4 regions from each of these 60 patients, so about 240 samples. And then the DNA and the RNA were extracted separately from each of these regions and profiled separately. So we used, for the RNA profiling, we used the NanoString pan-cancer progression panel, which looks at 770 genes. We also performed DNA sequencing using next generation sequencing using 225 genes. And we also performed multiplex ligation analysis, looking at gene amplifications of 16 genes. And essentially what we found from the genomic profiling of these regions is some very interesting findings. First and most importantly was that when we compared, and because these were all samples, regions from the same sample, we could actually perform pairwise analyses. And what we found was that when we compared the superficial aspects, because if you remember, we actually divided the superficial aspect of the tumor, or the mucosal aspect of the tumor, into 2 regions. When you compared these 2 regions, we actually found almost no genes that were significantly different, differentially expressed between the 2 superficial regions. And the reason we performed this analysis was because very often the surgeons or the gastroenterologist would say that, oh, the gastric cancer will be a heterogeneous tumor, and therefore we'll take multiple biopsies. And by taking multiple biopsies, we will try and encompass the heterogeneity that we see in the tumor. But what we found is, at least from a gene expression profile point of view, that we didn't actually see much expression differences between the superficial aspects of the tumor. However, when you compare the deep aspects of the tumor with the superficial aspects of the tumor, you actually see that about almost half these the genes were differentially expressed. And interestingly, that what we found was that when you compare the metastatic lymph node with the deeper aspect of the tumor, or the serosal aspect of the tumor, lesser number of genes were differentially expressed. And so what this analysis essentially suggested was that first is that the transcriptomic profiles of the superficial aspects of the tumor are actually very similar, but the superficial aspect and the deeper aspects of the tumor are very different, and that the lymph node metastases are actually more similar to the deeper aspects of the tumor, suggesting, number one, is that that just superficial biopsies may not actually encompass the entire heterogeneity that is seen in the tumor, but it also explains a little bit about the cancer biology, where it suggests that most gastric cancer tumors are actually starting from the superficial aspects and then metastasize through the serosa into the lymph nodes. And while this is something that sounds intuitive and has been shown in the Vogelstein model in colorectal cancer, for example, what has probably never been shown before is that this was performed in the same tumor. So in the Vogelstein model, for example, this concept of the tumor starting in the mucosa and then metastasizing to the serosal aspects, and then the lymph nodes were shown across multiple different tumors, but not in the same tumor itself. And so what we found was that the genes that were overexpressed in the superficial aspect of the tumor were actually quite different, and there were therapeutic targets such as CEACAM5, CEACAM6, CDH1, as well as FGFR that were actually differentially expressed in these different aspects of the tumor. All right, so this was, these findings were actually published in Gut last year, and the journal Gut last year. And moving on, we then performed another study, and this was focusing more on single cell sequencing of gastric cancer. So in this study, we took 31 patients with gastric cancer across all the stages, including distant metastases. There was a good spread of patients with intestinal as well as the diffuse subtype of gastric cancer. And these are is based on the Lawrence classification, for those of you who are not familiar with gastric cancer. And for 29 of them, we performed single cell RNA sequencing using the 10X platform, which involves a single cell dissociation as well as barcoding, and then sequencing. In 4 of those, we actually generated organoids, which we then also subjected to single cell sequencing. And in total, we sequenced more than 200,000 cells. And in a subset of samples, we also performed spatial profiling using the nanostring genomics platform. And for those of you who are familiar with single cell sequencing, one of the first things we did is we did unsupervised clustering to understand the different cell types that were present within the samples that we sequenced. And by performing this, we identified most of the large groups of cells that we will commonly see in gastric cancer, which includes epithelial cells, endothelial cells, fibroblasts and stromal cells, immune cells such as the plasma cells, B cells, lymphoid cells, the T and NK cells, as well as the macrophages and the mast cells. But more importantly is beyond the big cell types, which we could have actually potentially gotten using bulk sequencing, what we identified was cell states, which means within the plasma cells, we actually identified 5 different types of cell states. Similarly, in the T cells, we identified 5 different types of cell states. And in the epithelial cells, we identified 7 different cell states. And I'll explain a little bit about why these cell states actually matter. So one of the advantages of performing single cell sequencing on a very large number of cells is that we were able to identify rare cell populations. And one of the cell populations that we identified was this population that had a mixed lineage expressing both PLVAP as well as RGS5, which are separately endothelial as well as stromal markers. And these cells that were expressing both these markers seemed to suggest that there were cells that were undergoing endothelial mesenchymal transition. So this is slightly different from epithelial mesenchymal transition, which we are quite familiar with. But endothelial mesenchymal transition is something that has been documented and reported before, where there are cells that are starting to acquire features of either they're acquiring vascular features, or even what might suggest vascular mimicry, where tumor cells are starting to acquire features that look like blood vessels. And next, we went on to look at the different subtypes of gastric cancer and looking at their different immune cell proportions. So in the intestinal type of gastric cancer, as well as the diffuse type of gastric cancer, what we found quite starkly different was that there was a much higher proportion of plasma cells that was seen in the diffuse subtype of gastric cancer compared to the intestinal subtype of gastric cancer. And so we then went on to perform orthogonal validation of these findings by performing immunohistochemistry of IRF4, which showed that there was indeed a higher staining of IRF4 in the diffuse subtype of gastric cancer. And we also then went on to look at what type of plasma cells were present within the diffuse subtype of gastric cancer. And what we found is that it was mainly the PL4 and the PL5 cell states that were more present in the diffuse subtype of gastric cancer. And what we found is that the PL4 and PL5 subclusters actually had an upregulation of CD69, which suggests that this is actually an activated type of plasma cells that was being seen in the diffuse type of gastric cancer. And actually, when we went back to look at the TCGA, we could also see that the percentage of plasma cells in the TCGA using bulk RNA sequencing, because the TCGA mostly done bulk RNA sequencing, also seemed to suggest that there were higher proportion of plasma cells in the diffuse type of gastric cancer compared to the intestinal type of gastric cancer. So then moving further, we went to ask specific questions on, are there specific gene expression programs that are being expressed within the tumor cells that seem to attract these plasma cells. So this was one of the advantages of doing single cell sequencing, is that we could look specifically at the epithelial cells and the genes that are being expressed within the epithelial cells. And very interestingly, what we found out was that there was a gene that was upregulated in the epithelial cells of this diffuse type of gastric cancer called KLF2. And KLF2 is actually a transcription factor that is being associated with plasma cell homing. And so and this was first reported in multiple myeloma, where KLF2 was actually upregulated in the multiple myeloma cells, which are we know are aberrant plasma cells. And so what we went on to do was to see whether KLF2 was truly upregulated only in the epithelial cells that had close proximity to the plasma cells. And so using the nanostring genomics platform, what we did is we identified regions of interest that had, so in this, if you can see, we used multiplex immunohistochemistry staining for CD138, which is the plasma cell marker, and CK for tumor cells, as well as SMA, which is for the stromal cells. And what we could see what we did is we identified regions of interest where you can see epithelial cells that were in close proximity to plasma cells, stained in purple here. And we compared the KLF2 expression of these epithelial cells within this region of interest compared to epithelial cells that were distant from plasma cells. So for example, in this region of interest where you can see there's hardly any CD138 staining, and therefore there were no plasma cells. And what we could show quite clearly was that it was only in these epithelial cells that there was a higher expression of KLF2 compared to the epithelial cells that were far away from plasma cells. And this brings brings to context the benefit of being able to do single cell sequencing in our spatial transcriptomics, because these were features that we would not be able to identify if we just did bulk DNA sequencing or bulk RNA sequencing, for example. Next, we went to look at the fibroblasts, and what we found was that we found three different types of three different cell states in the fibroblasts. And what we found is that this is essentially identifying cancer-associated fibroblast sublineages. And we found that there is this gene called INHBA, which is a part of the activin-inhibin module, which is also associated with the TGF-beta as well as the Wnt pathways. And that this INHBA was associated only with 2 types of fibroblasts and not with the third one. And so this now starts to give you some context of the benefit of identifying fibroblast subgroups, because these will then become potential therapeutic targets in the future. And so we then went on to perform orthogonal validation of our findings in the single-cell sequencing on the nanostring, where we again identified fibroblasts, cancer-associated fibroblasts using the SMA staining, and then we sequenced these separately to look at various features, and we could see quite clearly that there was an upregulation of FAP, which is a known CAF marker in the tumor fibroblast compared to the normal fibroblast. And so one of the questions that as a clinician or as a medical oncologist, many people ask is, hey, all this is well and good. You've identified some genes that are up and down, but how do you actually bring this to the clinic, and how do you actually make these findings or biomarkers into, how do you make these findings into biomarkers or into therapeutic targets? And so for the last bit of my talk, what I'm going to talk about is, I'm going to share with you a little bit about how we have identified epigenetic findings that we have moved into the clinic and show the clinical relevance. And so, sorry, just before I move away from this slide, so these findings that we did in our single-cell paper was actually published in Cancer Discovery earlier this year. And for the last bit of my talk, I'm going to talk about promoters. So promoters are genomic elements that are upstream to the transcription start site, and they actually receive input from other genomic or extragenomic regulators, such as enhancers, insulators, or even environmental factors that then activate promoters, which then activate transcription at the transcription start site of specific genes. And so in one of our initial studies looking at epigenetic profiling and promoters of gastric cancers, we identified promoters in gastric cancers that were being upregulated compared to the normal. And these are what we call as alternate promoters. And what we found is in these alternate promoters, that a lot of the alternate promoters were actually downstream to the canonical promoters. And so the hypothesis here was that tumors were upregulating these promoters in the alternative transcription start sites in order to truncate the protein. And what we found is a lot of these tumors, a lot of these promoters that were alternative, actually, when we went to analyze which genes they were, we found that a lot of these genes were associated with high affinity binding to MHC class 1, suggesting that potentially these tumors were trying to truncate the N-terminal peptide, which we know is the most immunogenic aspect of the tumor, in order to achieve immune evasion. And so we came up with this hypothesis that there are actually 2 types of tumors. There are some tumors that have a regular utilization of promoters, but there are some tumors that have a high utilization of promoters called alternate promoter high. And these tumors, essentially what they do is that by altering the proteins and losing the N-terminal, the immunogenic N-terminal peptide, they cannot present to MHC class 1. And if they cannot present to MHC class 1, then there's no antigen presentation, and then these will not activate the T cells. As compared to the regular tumors, which should present to MHC class 1 and activate T cells, and then theoretically, these would be the ones that would be sensitive to immune checkpoint blockade, whereas the high alternate promoter utilizer tumors will be resistant to immune checkpoint blockade. And so in order to prove this hypothesis, one of the first things we did was we took a bunch of tumors that were treated with, of gastric cancer tumors that were treated with immune checkpoint inhibitors. And we studied the alternate promoter utilization within these tumors. And what we found was, indeed, there was this spectrum of promoter utilization across these tumors, with some having high promoter utilization and some having low promoter utilization. And it was interesting to see that the tumors with high promoter utilization actually had lower levels of T cell activity as measured by the gene CD8A, granzyme, and perforin. But even more proof of principle was the fact that when you looked at the survival plots, you could see very clearly that the high alternate promoter utilization had a clear resistance to immunotherapy with a very, very poor survival compared to the low promoter utilization. And this can be again seen in the swimmer's plot here, where the ones in blue have very good survival compared to the ones in red. In fact, this one tumor here in red actually is also microsatellite unstable, which may explain why this tumor responded well to immunotherapy. And so one of the things we then did was that most of the promoter activity that is measured with traditional platforms is using a technique called ChIP-seq, which looks at histone modification. And this is actually quite an expensive and difficult to do technique, which requires fresh tissue, fresh or frozen tissue. However, bulk RNA sequencing is actually done very commonly for most research platforms and technology, and is widely available, for example, like in the TCGA. So we developed an algorithm called ProActive that can directly infer promoter activity from RNA sequencing without performing ChIP-seq. And this was widely validated in multiple tumors in a pan-tumor analysis. And this was a separate paper that was actually published in Cell last year. But in this study, what we did is, using this algorithm called ProActive, we then applied the RNA sequencing. We applied this algorithm onto the RNA sequencing in over 10,000 RNA sequencing samples to generate what we call as the alternate promoter burden. And so we divided this into alternate promoter burden high, intermediate, and low. And we studied this across multiple data sets, including one of our data sets which we presented, which was the single cell sequencing study. And in this data set, we inferred the alternate promoter high and low for these samples. And what we could see quite clearly was that samples that were alternate promoter low had a much higher T cell proportion compared to the high and intermediate, again reinforcing our findings that these should have lower T cell activity. But what we then did is we then tested this in multiple data sets of immunotherapy treated cohorts of GI cancers. And in multiple cohorts, we identified we demonstrated that these high alternate promoter utilizers were actually having poorer survival, suggesting that this algorithm and this technique might potentially be used as a negative predictive biomarker for immunotherapy, selecting patients that are unlikely to benefit from it and then could potentially be treated with other alternatives. And this gives you an example of how this sort of epigenetic understanding of the tumor microenvironment epigenetic understanding of the tumor and the microenvironment will allow us to identify biomarkers, which essentially our group is now trying to develop this as a clinical test that can be deployed into the clinic now. And this could be used as a biomarker to select for patients who benefit or don't benefit from immunotherapy. And at the end of the day, the study of the tumor, interaction between the tumor and the microenvironment, components such as immune cells and stroma are going to be critical for the development of the next generation of cancer therapeutics and biomarkers. And novel technologies such as single cell and spatial profiling, as well as epigenetic assays beyond DNA sequencing, will be needed to be employed to study this tumor microenvironment. And so going back to my idea of using of this paradigm that I brought to you about this idea that to kill cancer cells, to treat cancer, one needs to kill cancer cells. I now present to you the Niche 2 study, which was recently presented by Miriam Chalabi in ESMO in September, where by just using immune checkpoint inhibitors in microsatellite unstable colorectal cancer patients who would have otherwise typically gone for surgery, more than 95% of these patients were treated with just 2 doses of immunotherapy and achieved pathological complete response, suggesting that we might need to now start shifting our paradigm away from this concept of to treat cancer cells. To treat cancer, one needs to kill cancer cells. To cure cancer, one may need to just assist the body in eliminating it. And with that, I need to acknowledge many people who have contributed to the various studies that I presented here, particularly the patient and families who have magnanimously contributed the tissue and clinical data to further the science. My mentor, Patrick Tan, whose lab I work in, and who most of the papers that I've presented to you are from, and of course his group. And of course, I would also like to especially acknowledge Heike, who worked with us on many of these papers that I presented today. And with that, I'll end my talk, and I'm happy to take any questions. Thank you.
That's a fantastic talk. Thank you very much indeed, Raghav. I really enjoyed that talk. Thank you. I'd like to kick off the questioning session, if I may, with a question of my own. I was very interested in your concept of the AP high cancers, and my question is, have you been able to show that AP high cancers do indeed have a reduced presentation of N-terminal peptides on the MHC class 1 molecules?
Thanks, that's a great question, and it's not that simple to do this. And so one of the things that we are doing is we have done TCR sequencing and on a bunch of cell lines, and we're looking at the antigen presentation of this. But cell lines are not that easy to bring back into the context, into the basic, back into a heterogeneous tumor, for example. The other place that we have done TCR sequencing is also in our single cell data set, which we are looking at now. So I'm hoping that we can answer some of these questions that you have asked. But one of the problems with the alternate promoter utilization is that to infer that from RNA sequencing, you can only infer it from bulk RNA sequencing and not single cell sequencing, because the depth of sequencing that occurs in single cell is not good enough for us to infer the different isoforms and therefore the promoter utilization. And so in that paper that I presented to you, we actually did both bulk as well as single cell to identify the promoter utilization. So this is a non-trivial step that requires a bit more fine-tuning to do. So the short answer to your question is we're working on it, but I hope to present the data in the near future. So yeah.
Okay, great, thank you. Now we do have some questions appearing in the Q&A box, so I will ask some of these questions, and I can also encourage audience members to think of questions and type them into the Q&A box, please. So the first question is from Asha Doob, who asks, do tumours with high alternate promoter activity still show positive PD-L1 staining on immunohistochemistry slides, or is the score reduced because of the lack of T cells?
Great question. I think, okay, so to give you some context is PD-L1 immunohistochemistry is only started to be recently done. It's not something that we have done in the past. So there are actually very few samples. And in fact, this has been something that I've been working on for the past 2 or 3 years, is to try and get to do RNA sequencing on immunotherapy treated or new era gastric cancer samples, which has been non-trivial. So what we have done is we've looked at PD-L1 expression from gene expression from RNA sequencing data. And we know that for sure there is lower expression of PD-L1, but it's measured from RNA sequencing and not immunohistochemistry. So that's the answer to that, yeah.
Okay, thank you. That's a good answer. So next question is from James Morris, who says, you mention extracellular vesicles in one of your slides. Could you enlarge on that, please?
Yeah, so that's actually, the extracellular vesicles is more as a review on the tumor microenvironment. And one of them would be exosomes, for example, which are being studied by multiple groups. We know that exosomes are something that carry small bits of either DNA or RNA or proteins, and have valuable information that can actually inform on various aspects of the tumor that by just looking at a certain protein or certain genes may not answer, but that's kind of where that space is moving. It's still quite early days in most of this area. I personally haven't studied extracellular vesicles myself. That was more a review of looking at the microenvironment that was mentioned there. So yeah.
Okay, so James asks a follow-up question, and he's really asking, is the study of extracellular vesicles, in your view, a good way of assessing information flow in the tumour microenvironment?
Great question. Again, not my area of expertise, but there are a lot of groups, both in Singapore as well as I know in other parts, including the UK, that are studying these extracellular vesicles. I think one of the problems that is going to arise with this, and this story is going to be very similar to microRNAs and long non-coding RNAs and all these other things, is that when you study this in a specific data set, is how replicable and easy it is to study the same thing in another data set, because that forms the basis of a biomarker development. And we have had this problem even with the alternate promoter utilization, which is from RNA sequencing. And a lot of us know that RNA sequencing is something that is not that easy to replicate in the clinic, which is why, again, as pathologists, most of you know that we always fall back onto a single protein with immunohistochemistry as something that's very easy to replicate. And one of the issues I will think with the extracellular vesicles is going to be this sort of thing. To bring it to the clinic is going to be the difficult step that one will need to do, and that will require large studies with large data sets, which I hope that we are trying to collaborate globally to try and pool some of these data sets to study this in a more definitive fashion.
Yes, thank you. Okay, so next, Adrienne Flanagan asks, do you ever see a cancer diverging and showing both AP low and AP high?
Great question. I won't be able to answer that because at the moment, the way we study alternate promoter utilization, again, I explained this a little bit earlier, is because you need to do it you can only do it on bulk RNA sequencing. To answer your question, either we have to do it like the first study we did with Heike, which essentially take the tumor, divide it in half, sequence them separately and ask that question. I haven't done that yet, but it's a good question and I might consider doing that. But from single cell sequencing, for example, I can't look at promoter utilization because the depth of sequencing in the RNA is not deep enough to look at isoform-based reads, and we only look at gene expression reads, which is not good enough for promoter utilization. However, to add to that, one of the things we are starting to look at, and we have done, is we've looked at copy number variation. So even though we've done RNA sequencing, we have platforms that can look at copy number variations within the single cells. And what we can and have shown quite clearly are that there are copy number variations within the same tumor, which means that there are certain cells and certain cancer cells that have a different copy number variation compared to the others. And this, we are writing a paper on this now, is actually looking at different mechanisms of tumor cell evolution, where some go through a branched pattern of evolution, whereas there are some that undergo punctuated evolution in their cells. And if that happens at the DNA level, I will not be surprised if that happens at the epigenetic level as well. And that's kind of my take on this. So,
yeah, okay, thank you. That's a good answer. So next question is from David Hughes, who says, excellent talk, thank you. In the study, where do you look at DEGs between different tumour areas and lymph nodes? Have you done pathway analysis to determine if the tumour in lymph nodes similar to the deep aspect of the tumour, or are you just relying on the number of the individual genes that are differentially expressed?
Yeah, very nice question, thank you. So so to give you some context and explanation for the panel. So number one is, at the time when we performed that study, we were not very confident. So firstly, we only had FFPE tissue. We didn't have fresh or frozen tissue from those samples. And so at that point of time, we were not very confident about doing whole transcriptome RNA sequencing on those samples, which is why we did the nanostring platform, which is a little bit more stable and validated for looking at RNA on FFPE tissue. But the problem with our nanostring is it is a probe-based analysis of the RNA expression, and therefore there's only a select number of probes that you can select, and typically for a nanostring, it's around 700 to 800 genes or probes. And so at that point of time, when we designed the study, there was a panel called the Pan-Cancer Progression Panel, which selected 700 or 800 genes that were cancer-associated genes, and this was what we studied. There was very little non-cancer related genes that were involved. So there were no immune genes, there were no other genes like or any of the other pathways that were non-cancer related that were included in this panel. But of course, within the 770 genes itself, there were some pathways that these genes were categorized into certain pathways, fertility, metastasis, angiogenesis, and those sort of things. And yes, there were clear pathways that were differentially expressed between these. But you must remember that we did a pairwise analysis of this, and that resulted in quite heterogeneous results in the pathway analysis, which is why we decided that since we only did 700 genes, that we might as well look at individual genes rather than pathways because of the limited panel that was being performed.
Great, thank you. So Matthew Brown asks, there seemed to be more of a difference in gene expression between the deep gastric tumour and the metastasis than between the superficial tumour and the metastasis. Not sure if he's right about that. Is there an explanation for that, or did I misunderstand the slide?
No. Okay. So when we compared the superficial layer of the tumor and the deep layer of the tumor, about 43% of the genes were differentially expressed. When we compared the superficial layer of the tumor to the lymph nodes, there was 38% expressed. So yes, the superficial layer seems slightly less differentially expressed to the lymph node compared to the deep, but I would actually approximate 43 and 38% as the same, which means that the superficial layer was quite different from the deep and the metastatic lymph node. But when you compared the deep aspect of the tumor and the lymph node, there was only about 16% difference, which means there's very little differences between the deep aspect of the tumor and the lymph node. So the conclusion from that is that the mucosal or the superficial layer is very different from the deep and the lymph nodes. That was the conclusion. So I think the way the numbers were, it's a little bit confusing, but that's the conclusion.
Okay, thank you for clarifying that. That's very useful. So your good friend and collaborator, Heike Grabsch, says, excellent talk, Raghav. Thank you very much. Do you think there will ever be a treatment for gastric cancer patients with all this heterogeneity we see even in individual patients?
Thanks, Heike. So I think that's kind of why I brought back that last slide on the niche 2 sort of thing, which the answers, unfortunately, to sort out the heterogeneity, we will need to rely on the intelligence of our own immune system to address that problem. It's going to be unlikely that targeted therapies are going to sort out the intratumoral heterogeneity. Chemotherapy, if you think about it, is also a form of targeted therapy because it's targeting one cancer cell pathway, basically. Whereas with immunotherapy, and here I'm not talking about checkpoint inhibition as a solution, because we know that checkpoint inhibition in gastric cancer only addresses some problem. But the pipe dream is that we have a cocktail of different drugs that are targeting different aspects of the tumor and microenvironment sufficiently to wake up our immune system to actually just take care of the tumor itself. And I think that's going to be the only way in which gastric cancer is going to get sorted out. But this is definitely a pipe dream. It's going to happen hopefully in my lifetime, but there's a lot of work to do. But in the immediate future, I do see that a combination of immunos with targeted or chemo is going to be the way to go for sorting this problem out. It's not going to be like lung cancer where but even in lung cancer, like a single drug to sort out a single aberrant oncogene is not going to be the way to solve this problem.
Sure, yes. Thank you. Okay, so Heike also comes back with another question, and it's in an area where I had a question as well. So I'll combine my question with Heike's. I was going to take you back to your data in diffuse gastric cancers where you showed changes in the plasma cell there, that you showed an increase in the PL4 state and the PL5 state, and you showed an increase in CD69 indicating activated plasma cells. And then further you went on to show an increase in KLF2, the plasma cell homing mechanism. So Heike and I guess, we're both asking a similar question. I'll read out Heike's, which is the intriguing finding of having more plasma cells in diffuse type gastric cancer. Can this be explored for therapy?
So I'll give it. Heike and me have been talking about this plasma cell story for a little bit now, and I mean, from a pathologist point of view, what I understand is that actually plasma cells are pretty easy to see, and we were a little bit surprised that this report of the diffuse type of gastric cancer having more plasma cells is something that, as pathologists, somebody would have seen it and reported it before we did it from a genomic point of view. And we were wondering whether this may not be a very strong result. But recently, I actually spoke to a couple of other groups that have also been performing single-cell sequencing of diffuse gastric cancer. They are finding exactly the same story about seeing activated plasma cells being more highly present in a diffuse gastric cancer. And so I'm now beginning to think that this may actually be something real. The truth is, our study and understanding of B cells in cancer in general has been quite weak. We've just about started to do it. I've been speaking to different groups, and almost nobody is doing B cell receptor sequencing. Most people have only been doing T cell receptor sequencing. And from a pathologist point of view, and maybe Mark and Heike can actually opine on this, is I'm just wondering whether what we are defining genomically as plasma cells based on marker genes that are more highly expressed is not something that is looking phenotypically like a plasma cell in the sample. Is that even possible, that they don't look like plasma cells, but they are marking like plasma cells genomically? They are highly expressing CD138, they're highly expressing IRF4, but morphologically they don't look like plasma cells. And is that possible? So is this what we did go back to look at is we did CNV analysis on these plasma cells, and we know that they're not aberrant. There's no aneuploidy in these cells. So that means these are not cancer cells. These are not cancer cells behaving like plasma cells, for example. That we're pretty certain. Heike asked me this a while ago. But are these immune cells broadly, if you will, that are looking like something else but behaving like plasma cells? And that's something that I don't know, and we're studying this. But given the fact that there are other groups now seeing the same finding, I'm more interested in this, and I'm going to answer that. But to answer your question of can we target this, the answer is yes, because if you now have we have identified KLF2 as a homing mechanism for the plasma cells. We know that the plasma cells are getting there, but they're doing nothing. I mean, they're clearly doing nothing. They're just sitting there. So the question is now, can we do something? This is where I think the new era of drugs that are coming into the market are going to answer this question, because typically KLF2 is a transcription factor. There's nothing we could do. There's no kinase or subunit that I can block using an antibody for this. But with the new RNA therapeutics that are emerging, with the new cellular therapies that are emerging, the hope is that we are now devising transcription factor disruptors with synthetic microRNAs, for example. This is something that BioNTech's actually pushing quite hard with their microRNA technology from their vaccines. And if you can start disrupting these interactions, these are protein-protein interactions from another angle rather than from a kinase driver gene sort of perspective, then I believe that this is the way that you will start to target these. And we will need to perform more studies. And again, this is very, very early discovery work that we've done. It's going to take a lot of steps to bring this discovery to the clinic. But this is the way I envision that the therapeutics are going to start being developed in the space, is with the new therapeutics that are not dependent on a kinase, for example, for targeted therapy, by disrupting these protein-protein interactions that we are finding upregulated in very specific cell states, that we are going to be able to target this.
Thank you very much for that answer. That was a very thoughtful and well-considered answer. That let us see some elements of your thinking, which was very interesting indeed. Thank you. That brings us to the end of the session. We have a tradition of finishing on time. It's 10 o'clock exactly now, so it just leaves me to thank Raghav very much indeed for a fantastic lecture and great answers to a very interesting set of questions at the end as well. Thank you very much indeed, Raghav. And that's the end of the session, everybody. Thank you.
Thank you very much for inviting me.
Thank you.